Hi, dear children. Welcome to the next session on the chapter Biotechnology Principles and Processes. Let us learn about the processes which is involved in recombinant DNA technology. What are the processes? The first is isolation of the genetic material or the DNA with the specific gene of interest, cutting the DNA at specific locations, amplification of the gene of interest using PCR or polymerase chain reaction, insertion of the recombinant DNA into the competent host cell, and finally, obtaining the foreign gene product and the downstreaming process of that particular product. So let us learn each step in detail. For isolation of a genetic material from a cell, the cell has to be treated with specific enzymes. And the type of enzyme which is used to treat that particular or to separate or isolate the DNA is specific according to the cell which is selected. For example, if it is a bacterial cell, the enzyme which is used is a lysozyme. If it is a plant cell, it is the enzyme cellulase. And for the fungus, it is the enzyme chitinase, which helps to break their cell walls. And after the cell is broken, the DNA along with other macromolecules like RNA, proteins, polysaccharides and lipids are released out. After breaking of the cells and releasing of DNA with other macromolecules, the RNA which is present is removed by treating them with specific treatments like treating them with our ribonucleases. And protein is removed by treating them with proteases, and other molecules are also removed by specific treatments. And after that, the DNA, extracted DNA is treated with chilled ethanol, chilled alcohol is added, and when the chilled ethanol is added, the purified DNA, we can see the purified DNA, it precipitates out as fine threads in the suspension. So the separated DNA can be removed by the step called as spooling. So this is spooling in which the DNA which is precipitated in its purified form by the addition of chilled alcohol. See, addition of chilled alcohol, the DNA starts to precipitate. And we can see this is a picture which shows the precipitated DNA, and the separated DNA is removed by the process called as spooling. Second is cutting of the DNA at specific locations. What are these locations and who cuts these the DNA? We know there are specific enzymes called as restriction endonucleases which cut the DNA at specific locations, and these locations are called as the recognition sites. So purified DNA is incubated with restriction enzyme. As a result, the DNA is digested. Digested means it is broken down into fragments. And the broken down fragments, it has to be separated. And we had learned in the chapter molecular basis of inheritance, we had learned how the DNA is separated in DNA fingerprinting. They are separated by a technique called as gel electrophoresis. And this agarose gel electrophoresis is employed to check the progression of the restriction enzyme digestion, how far the restriction enzyme has digested the DNA, and this can be checked by using the agarose gel electrophoresis. DNA is negatively charged. We know the charge on DNA is negatively charged, so it will automatically move towards an anode when electric charge is supplied. So DNA fragments are separated according to their size through sieving effect of the agarose gel. See, in this gel electrophoresis, the gel is made up of agarose gel, and this agarose gel is a polymer from seaweed. Okay, and the fragmented DNA, they are separated according to their size, through the sieving effect. This agarose gel is having many number of sieves or pores. So based on the sieving effect of the agarose gel, the fragments are separated based on its size. And in this gel electrophoresis, we can see the larger fragments are towards the towards this end, and the smaller fragments are towards the opposite end. And this process is repeated with the vector DNA also. So the DNA with the gene of interest and the vector DNA undergoes the same process, and they are separated using gel electrophoresis. And after the DNA are separated, they can be seen as bright orange colored bands. The separated DNA, they can be seen as bright orange colored bands when they are stained with a specific stain called as ethidium bromide. The stain ethidium bromide helps the fragmented DNA or separated DNA to be viewed as orange bands, and when they are exposed to with UV radiations, when they are treated with ethidium bromide and then they are viewed under UV radiation, the fragmented DNA in this picture can be seen in the form of orange bands. So this is the method by which the gel electrophoresis helps in separation of the fragmented DNA, and we are able to view that fragmented DNA. The DNA bands are cut out from the agarose gel. Now, where is the DNA fragments present? They are present in the agarose gel. This is the agarose gel, which shows migration of the undigested. You can see there are 4 lanes over here. 1, 2, 3, 4. These are called as wells in which the incubated DNA with the restriction endonuclease is poured into. And in the first well, that is the first lane, we can see the DNA is undigested. It has not been broken down into fragments. And in the second lane, third lane, and fourth lane, we can see all the DNA which has been fragmented, and they are moving through the agarose gel from the cathode to the anode based on their size. Okay. And its DNA bands which is present on the agarose gel, they are cut out, and this process is called as elution. The process of cutting out the DNA from the agarose gel is called as elution. And the cutout gene of interest from the source DNA and cut vector, they are mixed. Okay, so the gene of interest which is cut by elution and also the vector DNA which is cut by elution, they are mixed with the next enzyme, that is ligase, which is acting as a molecular glue, and thus we get a recombinant DNA. So this is gene of interest and this is a vector DNA which are incubated with the enzyme ligase, and thus we get a recombinant DNA. So this is the second step, that is cutting of DNA at specific locations. So dear children, in this module we learned about the first 2 steps involved in the processes of recombinant DNA technology. That is, first is isolation of the DNA with a specific gene of interest, and then separation of that DNA along with the vector DNA using gel electrophoresis, and cutting off that gene of interest and the vector DNA from the agarose gel through a process called as elution, and creation of a recombinant DNA. Thank you.